Welcome to our senior secondary biology. So in this video we're going to continue with genetics. So far we have looked at introduction, we looked at variation. In this video we're going to take a look at cell division. Then in the next video we'll look at monohybrid crosses. Then we'll end up finishing with blood group, sex linkage and mutation. So cell division or nuclear division. So nuclear division is another word for cell division. The reason why cell division is also called nuclear division, it's because the division or the dividing of a cell begins from the nucleus. So from the nucleus, the cell separates into two halves, as you are going to see later. That's why it's sometimes called nuclear division. So nuclear division is defined as a process by which new cells, which are called daughter cells, are formed from existing cells, which are called parent or mother cells. So there are two types of nuclear divisions. They are mitosis and meiosis. So one is called mitosis, the other one is called meiosis. We're going to start mitosis and meiosis in details. We start with mitosis. What is mitosis? Mitosis is a type of cell division that results in the formation of two daughter cells that are diploid and genetically identical from one diploid parent cell. Wow! So I hope we're able to understand this. We understand what diploid means, and of course genetically identical, it means they have the same genes as their parents. Okay. Yes, they are genetically identical, meaning that they have the same genes as their parents. With that being said, this is an illustration of mitosis. So let's say of this cell, it has 2N chromosomes, which is the diploid. So it's going to divide to form two, which have 2N also. And you can see they have the same number of chromosomes. So one diploid mother cell divides into two. The two are genetically identical. The two genetically identical. The two are genetically identical. And they are diploid daughter cells. So I was just disturbed a bit. With that being said, we go to stages of mitosis. So mitosis, we know it's where 2N, a parent with 2N produces daughters with 2N. But what are the stages? So this is where it gets a little bit deep into what happens in the cell for new cells to be formed. So there are four stages of mitosis. There are prophase, metaphase, anaphase, telophase. Here is a memory aid in case you want to master them. PMAT. P for prophase, M for metaphase, A for anaphase, T for telophase. So there is an organelle which we did not study in cells called a centriole. So a centriole is an organelle that is only active during cell division, and its function is to produce what are called spindle fibers, which are very important for cell division. So a centriole becomes active only when there's cell division. When there's no cell division, it's not active. So what happens during the four stages of mitosis, and why is the centriole so important? So in the first stage called prophase, here is what happens. So first, this is the cell, this is the nucleus. Of course, it has the nuclear membrane and of course the nucleus on the center. So inside the nucleus, that's where the centriole is. So these are the two centriole. So they become active only when cell division is about to happen. So what happens during prophase? So these two, what they do is they will migrate to the opposite poles of the cell. The poles of the cell are these two ends of the cell. So once they do that, the next thing that's going to happen is they are going the nucleus, the nuclear membrane and nucleus are going to disappear. When they disappear, then there's going to be what are called spindle fibers, which are going to be formed. So the spindle fibers are going to emerge like this from the centriole. So that's the role of the centriole. So to produce spindle fibers. So once spindle fibers are formed, the next is the chromosomes are going to shorten and coil, and they'll become visible. Usually they're invisible, so they are going to become short, and of course they are going to coil so that they become visible. So these are the chromosomes. which have become visible during prophase. The next stage is metaphase. So in metaphase, when all this has happened, what's going to happen is the chromosomes are going to migrate to what is called the equator of the cell. So let me just draw a cell here. So the chromosomes are going to migrate to the equator of the cell, which is the center of the cell. So when they migrate, the spindle fibers are going to attach themselves to the centromere, which is the middle of the chromosomes, of all the chromosomes. So this is attached to the spindle fibers attach itself to the centromere. This spindle fiber has done the same, this spindle fiber has done the same, and this one. So this is what happens during metaphase. The next phase is anaphase. So in anaphase, the spindle fibers are going to break and they are going to pull the chromosomes, breaking them apart. So what you're going to have is a situation, sorry, I did not thought I'd done a wrong illustration, but everything is okay. So the spindle fibers are going to break, and when they break, they are going to separate the chromosomes from the centromere in between. So when we have this, what we are going to have is a situation where the chromosomes are going to be pulled to one end, and these chromosomes are going to be pulled to one end. And the final stage in this dance of mitosis is telophase, where the cell divides. So when these are pulled from two ends, the cell is going to divide here. When it divides, then we're going to have two cells like this. The chromosomes again are going to coil, and of course the nuclear membrane and the nucleus are going to become visible again. So I've got two cells from one cell, and the number of chromosomes have been shared equally. So this is what happens during mitosis. So what are some of the importances of mitosis? First, growth. Second, regeneration. Third, asexual reproduction. Fourth, maintaining genetic stability. So let me just go through. I was just saying them without explaining some of them. So the first is growth. Because of cell division, when, for example, you cut your fingers, your nails, your fingernails, they're going to grow back because of mitosis. The second is regeneration, which is something that which is near the same. Let me just say that again. So growth is just normal growth. Even for the example that I've given, it happens because of mitosis. So for you to grow, the cells have to keep on dividing, making more cells so that you can grow in height and in width. Then regeneration is growing something that has been destroyed or that has been cut off. So there are some cells that can regenerate. So for example, let's picture a hypothetical situation. You have been cut, your hand has been cut, then it grows back. That is regeneration. There are some organisms capable of that. Then of course, asexual reproduction. So there are some organisms where there is no male or female, so the way they give birth is by dividing themselves. Then also maintaining genetic stability. So mitosis makes sure that the genes in a parent are the same as the ones in the daughters. So because of that, it makes sure that genes are not disturbed. So it maintains genetic stability. Because when genes are disturbed, when there is no stability, then we can have things or can have characteristics that we don't expect and which cannot cope with the environment. So it's important to have genetic stability in certain circumstances. Then of course, replacement of cells, which is more like regeneration a bit. But of course, if cells, when the more we do things, we end up using cells, and cells become old, they die. So those cells need to be replaced. So for example, you are hawking, there are some cells which you are killing by hawking, and when they are dead, they have to be replaced. So it's because of mitosis that that it is possible to replace cells, because the neighboring cells have to just undergo cell division to create other cells of the same type. So the best way to remember this is by this memory aid, Mr. Rag, where M is for maintaining genetic stability, R is for replacement of cells, this one. The other R is for regeneration, A is for asexual reproduction, and G is for growth. Next, we go to meiosis, or which is also called reduction division. So it is called reduction division because the chromosomes number, the number of chromosomes is reduced by half. So in short, if you have a diploid cell, then it undergoes meiosis, it's going to produce haploid. So it's called reduction division. So this is a type of nuclear division where one diploid parent cell produces 4 daughter cells that are haploid and genetically different from the parent cell. So meiosis produces 4 daughter cells. They are all haploid and they are not genetically identical as in mitosis. So if you have been asked to differentiate mitosis and meiosis, first it is mitosis produces 2, while meiosis produces 4 daughter cells. Mitosis produces diploid cells, while meiosis produces haploid cells. Mitosis produces genetically identical daughter cells, while meiosis produces genetically different um, genetically different daughter cells from their parent cell. So there are 2 divisions that take place during meiosis. So meiosis is a little bit longer as compared to mitosis. So it has 2 main stages. Each stage is made of the 4 stages that we have discussed from mitosis. So the 2 big stages of meiosis are meiosis 1, which is also called first meiotic division, and meiosis 2, which is called second meiotic division. So each of these stages are made up of the prophase, the metaphase, the anaphase, and the telophase. So this is a simple illustration of meiosis. We have one diploid parent. When she undergoes meiosis, there are going to be 4 children which are different from her. So, and they are all going to be haploid, of course. So this is an illustration of meiosis. So let's take a look at each of the meioses. Meiosis 1 and meiosis 2. What happens during meiosis 1 and what happens during meiosis 2? So remember, it's prophase, metaphase, anaphase, telophase, but there are just maybe 1 or 2 differences as compared to the prophase, metaphase, anaphase, telophase that takes place in mitosis. So we're going to highlight those. So meiosis 1, what happens? We have prophase 1. So prophase 1, what happens is movement of centrioles to the poles of the cell, just like in mitosis. The appearance of spindle fibers from the centrioles, just like in mitosis. Disappearance of the nuclear membrane and the nucleus, just like from mitosis. Chromosomes become visible by coiling and shortening, just like the prophase in mitosis. Then there is something that happens that doesn't happen during the prophase of mitosis. The homologous chromosomes pair up. What are homologous chromosomes? So homologous chromosomes are chromosomes pairs having the same genes in the same order along the chromatids. So there are some chromosomes that have the same locations of genes. Like you get this chromosome and these chromosomes, the types of genes and their locations are just the same. So such kind of chromosomes are called homologous chromosomes. So if 2 chromosomes are homologous, then they are going to pair up during prophase 1. Then there's something that's going to happen which doesn't happen in telophase, and this is very important, and it's called crossing over. So crossing over takes place between chromatids of homologous chromosomes. Remember, the chromatids are legs for the chromosomes. Then this crossing over, remember when we're talking about variation, what causes variation? We talked, of course, of the factors that contribute to variation, such as being controlled by many genes, and also effects of the environment. So this crossing over is another factor that, not variation, maybe diversity, genetic diversity. So why is it that we have different combinations of genes? So it is because of crossing over. So even variation, somehow crossing over contributes to that. So so what is crossing over? So crossing over is a process during which the chromatids of homologous chromosomes get entangled and exchange pieces. So I'll illustrate this so that you understand exactly what's happening. So this process contributes to genetic variation, like I said. So this is what happens. So we have these two chromosomes, okay? So if these two chromosomes are homologous chromosomes, which means that the location of genes on these two chromosomes are nearly identical, actually are identical. So if they are identical, what they are going to do is they are going to pair. So when they pair, they are going to be like this, and they are going to entangle or to intermingle to do some kind of H quad here. When they do that, what's going to happen is they are going to share the the ends of the chromatids, or the parts of the chromatids. So when they do this, this is going to break and it's going to join to this chromatid, and this chromatid is going to break and join to this chromatid. The same is going to happen down here. So what we're going to have is this kind of new chromosomes, where this has got part of the other one, and this has got part of the other one. So this is what crossing over is, and it is what also contributes to genetic diversity or variation. Diversity just means having different things uh, in a given population or sample. So after prophase, of course, we go to metaphase. So in metaphase, the same things happen as the metaphase in mitosis. Centromeres of homologous chromosomes line up along the equator of the cell. Spindle fibers from the centriole attach themselves to the centromeres of the homologous chromosomes. Then we go to anaphase, where the breaking happens. So during anaphase, one of the homologous chromosomes are separated from each other and put opposite poles, centromere first. So they are put apart. You remember the diagram had illustrated. So this is where something also interesting happens. So the chromosomes are separated randomly. So those chromosomes which line at the equator, they are separated randomly. It's not in a certain order. Because of that, the resulting daughter cells are going to be of different variety. So this random separation is called random assortment. So it contributes to genetic variation. Okay, so this random separation of chromosomes from the centromere, of course, when they are aligned at the equator, contributes to genetic variation, just like crossing over does. So these are the interesting points of meiosis: crossing over and random assortment. The rest is just like in mitosis. Fine, I'll go to telophase 1, where the chromosomes arrive at the poles, the spindle fibers disappear, chromosomes disappear by unwinding and becoming longer. The nucleus and nuclear membrane reappear. So I'm just talking about the point where the cell now finally divides to form new cells. Then in meiosis 2, what happens is exactly the same as in mitosis. So meiosis 2 is just mitosis. Okay, so of course the stages are called prophase 2, metaphase 2, anaphase 2, telophase 2, but in essence you don't need to know them, because if you know what happens in mitosis, the same happens in meiosis 2. So what are the importances of meiosis? One, formation of gametes. So sex cells are haploid. Where do they come from? They come from diploid cells, somatic cells that are diploid. So for us to have haploid from diploid, there is meiosis. It contributes to genetic variation due to crossing over and random assortment of chromosomes. So remember, crossing over, random assortment contributes to genetic variation. It's good to have genetic variation in a population. So these are the two importances of meiosis. So this marks the end of this part of cell division. I know it's a little bit involving, but I hope that illustration of mitosis can help you visualize the various steps. Otherwise, happy solving.